neurons get out from the cycle as we become adults. So I know that, for example, many cells, interneuronal cells, they are called, I don't know what they are called, but there are 5 types of interneuronal cells, and as we learn, as we do some things, they increase the number. But neurons, I think that they get out from the cell cycle and they do not regenerate.
So most of the brain in most of the brain we don't get new neuron formation once you reach adulthood. There's only, so far what we know, in about the last 2 decades, we've discovered that there are 2 regions in the brain that throughout our life we get the formation of new neurons. But what you'll learn today is when we get older, it basically becomes almost like nothing. So when you're in your 20s, when you learn, you're able to get new neurons. But then once you hit your 40s and beyond, it goes down. And these new neurons are able to integrate, but this only happens in special regions of the brain, not the whole brain. So I don't know if that answers your question, but most,
yes.
I mean, once you're post-mitotic neuron, you're kind of, neurons don't divide, but neural stem cells have the ability to, but only in discrete regions of the brain. Say again?
How does this stroke or epilepsy regarding?
So stroke and epilepsy will cause a lot of damage to the region in neuronal cell death. And because of certain growth factors that are released, it'll cause just a little bit of neurogenesis, although it's not enough to actually repair the brain. So there are ongoing studies on how we can manipulate the environment after stroke to induce new neuron formation in order to repair the brain. But in mammals, in humans, just a little bit happens, but it's not enough to actually repair the damage.
So that's the damage that is
It's not. There's kind of some damage after a seizure, and there's like immune cell infiltration, and then that can cause some cell death.
Epilepsy will lead to hypoxia when we get hypoxia, the oxygen will not send to the brain cells. After that, it will be degeneration of the cells.
Yeah, that's true. But then, as a result, the brain tries to repair itself, but it's just not good at it.
It's a training to the brain.
Yeah.
Not neurogenesis as this meaning when you are in before the puberty and after.
Yeah, there's always low levels of neurogenesis, but it increases transiently after a stroke, but not enough to actually replace the dead neurons. And so, as we know, so a neural stem cell has the ability to differentiate into a neuron, an astrocyte or oligodendrocyte. And what actually instructs the neural stem cell to differentiate into a specific cell type? What's important is the microenvironment in which it lives in, the cues around it. These include growth factors, extracellular matrix, small molecules, and as I said earlier, other cells like the endothelial cells, the astrocytes, and even mature neurons. So it's complex signals that can interact to regulate stem cell function. What I was interested in my thesis work was in understanding how growth factors and morphogens in the brain can regulate neurogenesis and how they change with age to inhibit neurogenesis, and if we can actually reverse that. And so, basically, on the left you see a young brain. You have healthy neurogenesis, neural progenitor cells, NPCs will become neurons. You have microglia, which are the resident immune cells of the brain, but because you're healthy, they're in an anti-inflammatory state. They're not doing much. My interest and my thesis work is in understanding first what happens with aging to the brain. How does neurogenesis change? Does inflammation change? And how can we then reverse that? And so in order to study this, I use the mouse as my animal model. So a 2-month-old mouse that you can see on the left has a lot of neurogenesis. So another technique I use in the lab to study neurogenesis is this chemical BRDU, which is a thymidine analog that when we inject into an animal systemically, any cell that's dividing will take it up instead of thymidine. It's like an analog of it. We can then use fluorescent microscopy, we'll section the tissue after we kill the mouse, look under the microscope and stain for BRDU, and whatever cells are positive, we know have undergone proliferation. In the brain, because most cells are postmitotic, like what you said earlier, neurons don't divide, if they uptake BRDU and they're in a specific location that we know neurogenesis occurs, we know that they're progenitor cells. So here in the young, what you can see in the yellow and red are proliferating cells, lots of progenitor cells. Then in an older mouse, so a young mouse 2 to 4 months of age is like a person in their 20s. An old mouse that's about 2 years old is like a person in their 80s. And as you can see, now suddenly there is almost no neurogenesis. There's about a 90% decline in the ability of the brain to form new neurons when we grow old. And so when I started my thesis, basically there was almost nothing known about why these cells stop working. Why do the neural stem cells die? 50% of them die off. The ones that remain in the brain, they won't become activated. They don't start proliferating upon cues, on learning or diet or exercise. And so that was my interest in understanding. Another thing that happens are the immune cells in the brain change when we get older. So in microglia in a young adult, basically they're pretty quiescent. They're surveying the area around the environment. Because you're healthy, there's not much debris, they're not doing much. When microglia in an aged brain, they become what we call pro-inflammatory. They start secreting more cytokines, which can then kill neurons. And even if you do have an infection, their ability to eat to a degree, like phagocytosis, becomes impaired. They become cytotoxic and chemotoxic. Exactly. And the reasons for this are still not really well understood. And so this is a picture in the hippocampus where neurogenesis occurs in a young brain. In green are the microglia. I stained them with a marker of microglia, and you see there's not that many of them. CD68 is a marker of lysosomes, so the more red you see, the more phagocytic the microglia are. When you get older, there's an expansion of the immune cells, and they become more reactive. You see more CD68 positive microglia. I'm interested in understanding both why that happens and how these now overactive pro-inflammatory immune cells in the brain can actually inhibit neurogenesis. And are there ways in which we can reverse this in old people? And so the first thing I'm going to talk about is work from my doctoral thesis on biochemical changes that occur in the microenvironment where the stem cells reside with aging, and how I've actually developed methods for rejuvenating neurogenesis and reducing inflammation. And so here I'm going to switch to a growth factor signaling pathway that is very important for regulating neurogenesis, and which I'll show changes with age. And that's colon morphogenic protein signaling. It's a member of the transforming growth factor beta family. So it's a cytokine that is a protein that's in the extracellular matrix that, when it binds its receptor on a cell, will induce a signaling cascade that results in phosphorylation and activation of these SMOD transcription factors, which then, when they translocate to the nucleus, will induce transcription of a variety of genes that control cell fate. In the case of BMP signaling, it actually will inhibit neurogenesis in favor of glial formation. And so what's known about BMP signaling? Why was I interested in it? Oops. So BMP signaling has been shown to inhibit proliferation of progenitor cells and maintain neural stem cells in a quiescent, undifferentiated state. It's also been shown, if you inhibit BMP signaling, to actually expand the stem cell pool in the hippocampus. And when you exercise, we know that exercise enhances neurogenesis. So everybody should go walking and exercise every day because it's one of the best things for enhancing neurogenesis. But one of the mechanisms by which it does is actually by inhibiting this pathway. BMP signaling has also been shown to increase in the brains of people with Alzheimer's disease. So I wanted to study it during normal aging. And so this is a study that I published last year for my thesis work. So I'm just giving you guys kind of the article if you want to read more about it later, but I'll show you some of the data where I show that there's an increase in BMP signaling with age, and it is because it increases in the environment around the stem cells, it inhibits neurogenesis. And how did I show that? So first thing I wanted to show is that it actually increases in the microenvironment by which the stem cells reside. So I did immunofluorescence staining, and in red you see in an old brain, so again this is from a mouse that's 2 years old versus a mouse that's 2 months old. In the hippocampus where the stem cells reside, you see this increase in red, which is more BMP signaling, which is actually the way it's shaped, you can tell it's in the blood vessels. The blood vessels are the ones expressing BMP. You also see at the mRNA level different family members. So BMP, there's different growth factors that are similar to each other and all signal through SMODs, but they're slightly different. There's different family members. So BMP4 and BMP6 at both the mRNA and protein level, I saw that it increased in the brain. You see more just staining in an old brain, and then even when I measured the mRNA that I isolated from the hippocampus through quantitative PCR, you see this increase. And then here you see an increase in the other family member, BMP2. Then what I did is I took neural progenitor cells and basically cultured them in a dish, just so I can really study how it works. And when I add this protein just to the stem cells in a dish, in vitro we call it, you see when I also then add the chemical BRT to label proliferating cells, a 50% decrease in the amount of proliferating cells. To confirm that I'm actually inducing the signaling pathway, I isolated the protein from the progenitor cells, and you see that the phosphorylated transcription factor that's downstream of the protein goes up. So basically I add it, it induces this pathway, and as a result you get inhibited proliferation. So I'm just showing you guys bits and pieces so I can show you a lot of different stories. So I'm skipping a lot of data, but now I'm going straight to then asking the question, will directly inhibiting BMP signaling in the hippocampus of an aged mouse lead to an increase in neurogenesis? So it's not enough just to see that it increases. I actually have to prove that if I inhibit it, I can enhance neurogenesis to show that that is one of the reasons why we no longer have neurogenesis when we get older. And the key goal of this is in understanding ways to rejuvenate the aged environment around the stem cells in order to enhance stem cell function. So a key kind of theme is that throughout our body, in the brain and even in other tissues, until we die, we have stem cells. The issue is because the environment around the stem cells change, it becomes more inhibitory, they're no longer able to become activated and to regenerate tissue. So if we can find out what exactly is causing the changes in the environment, we can use medicine, inhibitors, things like that, to reverse these changes to more youthful levels in order to activate the stem cells and regenerate tissue. So that's the goal. And so a technique I use in the mouse is viruses, like a gene therapy method, where I took a virus and I inserted in it an shRNA in order to inhibit the BMP signaling pathway. So viruses are often used as techniques both in just biology, to study biology, but also there are a lot of companies now trying to use viruses in order to fix genes in the body as a method of curing diseases. So I turned to try to use a virus. And what did I put in the virus? Oops. I inserted in the virus an RNA. And basically what the RNA does, the virus will invade the cell, go into the nucleus, and then the RNA that I encoded in the virus will be released. And what it actually does is if the RNA will bind to the gene of interest and prevent it from becoming a protein. It's a technique that we use in order to inhibit a gene of interest to study it in research. And so I took an old mouse and I actually drilled a hole into its brain and injected the virus with an shRNA against the transcription factor, SMAD1, which is downstream of BMP. So when I showed you before, the BMP protein signals to the receptor and then it activates the SMAD. If I can inhibit the production of the transcription factor, I can inhibit the pathway. So that's what I tried to do. I then gave the animal about 10 days to recover, and then I injected the animal with BRD to label proliferating cells. And what I wanted to see is, if I can see increased proliferation. And in fact, I could. So when I inhibited the BMP pathway through SMAD1's shRNA, I could see an increase, not too much, but a greater increase in these proliferating cells in the region where neurogenesis occurs. I got about a twofold increase in progenitor cells. It's still way less than what happens in young, but it's a good start. And so now I'm going to switch to a different pathway, another family member of the BMP signaling pathway, TGF-beta. And so, any questions so far? Yes. Okay, so I also studied TGF-beta signaling, which, like BMP, will bind to a receptor. It's located in the protein, in the environment around the stem cells, will activate the SMAD transcription factors, which then, when activated, actually induce what we call CDK inhibitors, which are these proteins, when activated, when expressed, will inhibit the cell cycle, so they prevent cell proliferation. And what do we know about TGF-beta signaling? We know that it inhibits proliferation. We know that it actually, we already know it increases in an aged brain in other regions, not the region of neurogenesis. And we know in the other region of neurogenesis that it's actually secreted by the endothelial cells, and that overproduction of TGF-beta is associated with neurodegeneration and many different cancers. So it's an interesting pathway to study because of its role already in other disorders and cancer. And so then here I'm going to show just a little bit of data from this study that was published last year on how if we can inhibit TGF-beta signaling systemically, I'm going to show you, through the blood, we can actually enhance neurogenesis and even enhance muscle regeneration, which also declines with age. So it's actually an exciting therapy for being able to, reverse aging in multiple tissues at the same time through the blood, which is going to then be the next theme of my talk. Yes?
Why do BMP and TGF-beta increase? Why do the cells start expressing those genes?
That's a great question. So why exactly does it even go up with age? We're not sure, but one possible reason could be that, throughout life, we constantly need protection from mutations from cancer. So some things might happen when we get older in order to protect the body from what we call oncogenesis, mutations, just because of the environment. So that could be one reason, is actually in some ways protection. Others could just be because of your immune system becomes overactivated when you get older, and immune cells might secrete TGF-beta. But it's not really we're still trying to understand, why do we age in general? That people don't really understand, but we're trying to get at that. So it's a great question. And so it was also shown before that TGF-beta increases in the muscle and can actually inhibit muscle regeneration by upregulating these CDK inhibitors, which inhibit the cell cycle. So already it was studied in muscle before I studied it in brain, in the lab that I was in. And so what I wanted to know also is if TGF-beta goes up in the muscle, is there similarities between muscle and brain? Because if you can find similarities with how stem cells are deregulated in multiple tissues, then you could use the same therapies to not only enhance brain function, to regenerate your muscle, to make your heart healthier, to regenerate your pancreas, your lungs, basically to just make you healthier as a whole. So I wanted to know, are there similarities? So to give you a little bit of background on actually muscle. So muscle, in our body, we actually have muscle stem cells. And where they're located is here, the satellite, they're called satellite cells, underneath the basal lamina of a muscle fiber. And so whenever you exercise or you get injured, basically what happens on day one, you can see like, what we do is we use a mouse. Again, the mouse is our model because you can't really do this in people. And so if we either exercise the mouse or even inject a poison to rip the muscle fiber, what you notice is 5 days later in a young mouse, you get the formation of these beautiful new muscle fibers, right? With the essential located nuclei. But in an old 2-year-old mouse, rather than getting new fiber, you actually get scar tissue. And this happens with people. So that's why when you're young, you go to the gym, you actually will get nice new muscle, you feel great. If you overdo it when you're old, you're going to just get scar tissue instead of muscle. Why? Because the stem cells, the muscle stem cells, are unable to become activated. But at least in a mouse, there's been many ways we've discovered on how to actually rejuvenate muscle regeneration through changing multiple pathways, like TGF-beta, inhibiting TGF-beta signaling, enhancing a notch signaling pathway. Through what I'll teach you, I'll show you later, parabiosis, when you connect the blood circulation of a young and old animal. Or we'll also show you later through adding proteins secreted by embryonic stem cells. Yes?
It sounds bizarre to me that there will be skeletal muscle regeneration because when we exercise, when we're doing something to strengthen more muscles, there will be a benefit for the muscle itself. I mean that there will be an increase in the size of the skeletal muscles, not increase in the
No, there's new muscle regeneration because what happens when you actually exercise is you tear your muscle. Also with age, we get what we call sarcopenia, which is just, loss of muscle mass, loss of muscle tissue. That also happens when we get older. But even the ability to make new muscle becomes severely inhibited, once you hit like 40s, 50s, 60s. It just, you get scar tissue instead it's because your stem cells won't activate to make new fibers, but you do get new fibers every time you exercise. New cells, you get new cells, new muscle fiber cells. مسية. You do. You get proliferation, and then you get then they become like tubes, and they form new fibers. And so I'm going to skip to kind of the highlight of the study. In this study, just like in the BMP study, I also injected a virus into the brain, and I saw that it could enhance proliferation of the stem cells. But what I want to know, is there a way, non-invasively, without having to put something directly into the brain, through the blood, that we can actually enhance neurogenesis and even enhance regeneration of muscle? And so I turned to a small molecule inhibitor of the TGF-beta pathway that's already now in clinical trials for treating cancer. And so we call this alpha inhibitor. So what I did is, over the course of 10 days, I injected every day this small molecule inhibitor into the bloodstream of the mouse. Then, 5 days before I killed the mice, I injected them with VRU to label proliferating cells, and I also injured their muscle with, it's like a snake venom, a poison that's what we use, to sort of cause the muscle fibers to die. And then, as a result, they have to make new muscle fibers. And so here is just how I'm showing that when I actually add the inhibitor to neuroprogenitor cells, you get an inhibition of activation of the downstream pathways. We know the inhibitor works. The first thing I want to do is see, does the inhibitor do what I think it does in inhibiting TGF-beta pathway. I also want to see, so you can imagine in the bloodstream, how are we going to do this? So if you inject anything in the blood, most drugs can't get into the brain. Why? Because of the blood-brain barrier. Is it got Yeah, small molecules that are lipophilic, they have to be less than, I think, 40. Very lipophilic and also very small. So most things can't get through. And because of that, that's why the brain is the hardest, tissue, the hardest organ to treat. Why is there still basically no cure for Alzheimer's disease? Because we can't get antibodies into the brain. A lot of cancers are hard to treat because you can't get drugs in. So the first thing I want to see is this is a small molecule. Can it actually, some molecules, if they're small enough, and like you said, lipophilic, can cross. So I changed for just to see if the molecule, if I inject it through the bloodstream, can it get in? And what I see is that when I inject it through the blood, it can get in because PSMA3 downstream of TGF-beta is inhibited. Red is like the signaling. It's actually active in most cells in the hippocampus. And when I inject the drug systemically, you get a lot of inhibition of that signaling. So we know that it crosses the blood-brain barrier. And so then, basically, what I did after I injected for 10 days the molecules, I looked at proliferation like I did before, and as you can see, in the region where neurogenesis occurs, there's about a 2-fold increase in proliferating cells in the brain. And this is pretty exciting because this is just injecting it, systemically. So it's something that might be applicable to humans. Then I looked at muscle fiber, and so in the muscle, so again, I injured the muscle before, while I was injecting the inhibitor, I also injured the muscle fiber with this snake venom. And so when I look, in the old mouse, the vehicle, you see barely any green cells. So green is a marker of the new myofibers, this myosin heavy chain protein. In an old mouse that just had the control vehicle, not the inhibitor, as expected, you see almost no green, no new fibers. It's mostly scar tissue. But when I added the inhibitor, while I injured the mouse, it regenerated way better, way better. So that was really exciting. And so another important thing to note is I only looked at brain and muscle, but maybe other tissues also had positive benefits from this inhibitor. So now there's a lot of interest actually in seeing therapeutically if this could be something we could use just to treat aging in general. Yes?
Were there any side effects to the
Great question. I didn't look long enough, but we'd have to do that. We'd have to see if, the mice, within the 10 days that I treated them, they were healthy, they were fine, they actually looked better. Because they get better muscle, better brain function, but I don't know if it's and right now it's in clinical trials for cancer. So far we don't think there's negative side effects, but we'd have to do more pharmacokinetic, pharmacodynamic studies in order to really see how it acts and how long it's in the system, and if you see any negative side effects.
So I thought a little bit of maybe impaired immune function.
It does the opposite, actually.
I know, but if there was some condoms or so, the use may increase in activity. So
That's
there
true.
might be some autoimmune disease or
Potentially. So actually, I'll talk more about immune system and regulation in a little bit, but I didn't show it here. But basically, what we see is that with age, there's impaired immunity, you get greater inflammation. And actually, when we add this inhibitor, it actually it decreases it a bit. And so that was also a big study. This I don't show that because I want to show you guys 2 other projects. But that's a great question. And so also, like I said, I also did the virus. I injected the virus in the brain, and I see increased neurogenesis, just like with the BMP signaling. And when I inject the virus to inhibit SMAD3, I also get enhanced muscle regeneration. So this is like kind of a different way of looking. Instead of just fluorescence, it's you stain the tissue, and you can just look at the different colors and see muscle fibers and then new fibers with central located nuclei. And you see more of it when you add the virus than the control virus. And in vitro, when I look, if I inhibit the TGF-beta pathway with the virus, I get increased myotube formation, or new kind of increase in proliferation of the muscle progenitor cells in a dish, as compared to when I just add the control virus.
It's not that you insert the VM into the virus and make a mutation in the genes to stop the signaling to the VM.
Yeah, well, this is TGF-beta. So how?
It is not a drug or a chemical.
No. So how does that?
Experimental, you take the PMB and insert to the virus and studying the gene and screening the map of the gene and you stop the signaling to it. And you insert to the mice, insert the virus to the mice in the location to generation, the cells themselves.
No. So the first, what I showed you is I injected a small molecule inhibitor, but I use multiple methods in order to prove my point that the pathway is important. So the small molecule inhibitor is something that can be therapeutically relevant because it's just a small molecule and it goes in the bloodstream and it goes everywhere in the body. What I'm showing you is, in addition, I wanted to show another way through genetic means that this pathway is important. In another set of mice, in a different experiment, I also injected the virus to inhibit the pathway. That would be less relevant as a therapy. It's more to study because it's a little dangerous to add a virus and cause mutations, things like that in the cells. So now I'm going to get to my current research, what I'm doing as a postdoc at Stanford. And before, what I was doing is I was studying the environments around the cells, the proteins, to see how they change with age and if we can reverse them, both locally with a virus and then systemically with a small molecule inhibitor. Now I'm interested in understanding how is it that the blood, our blood actually changes with age, and can that communicate with the brain to inhibit brain function? If we understand how it communicates, then we can change things in the blood to affect the brain and enhance brain function. So that's now my new focus. So in the last basically decade, we discovered through a really weird technique where we can actually sew together a young and old mouse in order to have them share their blood circulation. So a 2-month-old mouse with a 2-year-old mouse. So now they're sharing blood. And what we noticed is the following. We noticed that actually young blood in the old mouse can regenerate every single tissue in that old mouse. It can enhance pancreatic function. It enhances the heart. It creates new bone. It enhances skeletal muscle regeneration. And in the lab I'm currently in, just in the last 4 years, we discovered that it even enhances neurogenesis. While young blood is rejuvenating to the old mouse, the opposite effect happens in the old, where the old is actually inhibitory to the young and inhibits brain function. So I'll speak a little in detail. So when it comes to brain, we know that young blood can actually enhance myelination in the spine in like a mouse model of demyelination, so it might have positive effects for people with multiple sclerosis. We also know that the young blood, when in an old mouse, can enhance what we call synaptic plasticity. So it causes new dendrite formation, increased spine density in the neurons, increased potentiation, and causes the mouse to be smarter in cognitive tests. It also enhances neurogenesis. Like I said, the opposite is true of old blood. If you take old blood and inject it, either you do parabiosynthesis surgery to connect the mice, or even if you just take the blood and inject it, which I'm going to show you later, into a young mouse, you get decreased neurogenesis. Here, what's in brown is immature neurons. And what you can see is when you add old blood, this is systemically, you actually get about a 25% decrease in the amount of immature neurons and their intelligence, basically. If they can't hold independent cognitive tasks, they're stupider. So, and what are the mechanisms by which this happens? We're not really sure yet, and that's where I come in. So far, we know that there are some immune molecules in the blood that go up with age that contribute to this, like this chemokine CCL11 or beta-2 microglobulin, which is a component of MHC2 class molecules, maybe MHC1. But this, in general, goes up, and if you decrease these proteins, you can prevent the inhibitory effects of old blood. Yeah. But here's where kind of the question is. Like we said before, with the blood-brain barrier, most factors, cells and proteins in the blood can't get into the brain. So how can the blood be rejuvenating if it's young or inhibitory if it's old? How can it do anything to the brain if it can't actually get in? So my hypothesis, what I'm studying right now, is that it's not probably, most of them aren't getting in, they're probably acting through, what are they in direct contact with? Exactly, the brain endothelial cells. So that's now what the focus of my research currently is in understanding how do these endothelial cells in the brain change with age, how are they affected by the blood, and how might they mediate signaling from the blood into the brain. And the reason that's so exciting is if we can really understand this, we might be able to inhibit things in the blood to make the blood-brain barrier healthy, to prevent inhibitory effects in the brain, to enhance brain function, without having to drill a hole into the brain or to do surgery. So it's way more therapeutically relevant. And so the first thing I did was like a proof of concept that if you inject blood, you actually, systemically, just the plasma, not even the cells, just the blood plasma, you can inhibit young brain function. So over the course of 4 days, I injected young mice 2 times daily with either young blood, which shouldn't do anything because they're young mice, or old blood. So blood that I took from a mouse that is 2 years old into 2-month-old mice. And what I see here, then I also added ferritin like I've been showing you guys. And so in a young mouse that got young blood, you get lots of proliferating cells. But in the old, you get about a 30% decline. Each dot here represents a different mouse that I used. Then I decided to also look at the immune cells in the brain, because I want to know, if there's these, if old blood is more inflammatory, can that somehow cause more inflammation also in the brain? And in fact, it does. So what you see, what I showed you guys kind of earlier with the INA1CD68, in a young mouse, there's microglia, but they're not so many. They're pretty. They're not very active. There's not much CD68 staining, which is a marker of the lysosome. But when I add twice daily, just for 4 days, blood, and this is just injecting it systemically throughout the body, we're getting increased activation of the microglia, which suggests there's now more neuroinflammation. And so basically, what we see is that with aging, there's a decrease in neurogenesis and there's an increase in brain inflammation. So now, as I said earlier, considering that the blood-brain barrier is impermeable, how can factors in old blood inhibit neurogenesis and activate the microglia? How does it work? That's what I want to know. And so, in order to understand this, what I actually did is I just turned to actually now human blood from humans that are 25 years old, 18 to 25, versus blood from humans that are 65 and older. And we looked at all the proteins in the blood that they express through like a technique, like a microarray analysis. So we did proteomics, so you see the heat map, and what I want to see is what goes up with age that might be an indicator of change in the blood vessels, the vasculature. And so I came up with this protein, vascular cell adhesion molecule 1, which is actually expressed in endothelium throughout the body, and its shed form, the shed form of it, is going up with age. And so here then I actually took blood and I measured, through what we call an ELISA, I measured the amount of protein, this specific protein, and I see there's a lot of it in the blood with the shed form, and it goes up with age. I then, of course, went back to mouse to see if it's similar in a mouse. And what I see is in young mice, and middle-aged mice, there's the same level of this shed DKM1, but in an older mouse, it increases. I then performed the surgery where I connected the blood circulation of young and old mice. And what I noticed is the young mice connected to old blood had this increase in DKM1, while all the other mice, the older mice, remains high, suggesting that this is something that goes up with as a result of something in the old blood. So what is DKM1? Well, when you have increase in inflammatory, yes?
I think it's not the same like the including mice and like I said, the blood circulation through mice.
So if with young mice, it stayed low.
Yes, it's like the same. But it's just a unique regression on
So what we actually do is we surgically connect the bloodstreams.
I mean, for example, the circulation, like one, another one,
That's true. So each one of these, each dot represents a different mouse. Every single dot, that's why I like to kind of present my research instead of graphs. I now just show you dots. Every one is an individual animal. So, this one for some reason was really low. But in general, on average, it's around this level. And then each one of these is a different animal. So, you have some old mice that have levels almost as the same as the young. But on average, it goes up. So there is variability from animal to animal. But it's important to know that the mice that I study, they're all actually genetically identical. The mice in the lab that we breed, we make sure that their genomes are exactly identical so that there's not too much variation. But in humans, of course, there's a lot of genetic variation, but what we saw, what I showed earlier with the humans is still, there's pretty much an average low level in young people that goes up with almost all old people. But you always get, just like you see, like some people will age and they'll be super healthy, and they still have factors like, and their blood, if you even look at their blood, it's like they might be 80, but they look like they're still 30. We don't know really why, but there is variability from people to people. But in general, we're looking at things that increase with most people. Yes?
Did transferring blood affect the immune response in maybe cellular and
That's a great question. I really want to know this, and I actually collaborated with another lab at Stanford to look at the blood cells, and it's just so complicated. It's hard to, we see changes with age. Like, for example, T cells become more activated with age. You get an increase in the myeloid cells instead of the lymphoid cells, I think. You get changes, but when it comes to parabiosis, when you share the blood circulation, it's just too much, it's messy. There's too much variability from mouse to mouse to make any average. Because I did look at it, and you'll see why I looked at it. I'll explain. But we don't, so far, and I've spent a lot of time looking, I can't see any difference. And so what does BCAM1 signaling do? Well, when you get an increase in inflammation with age, like, for example, TNF-alpha, I'll probably need about 15 more minutes. Is that okay? 15? Okay. So when you get an increase in TNF-alpha, IL-1 beta, it binds receptors on the membrane, on the blood-facing luminal side of the endothelial cells, and uses signaling to upregulate adhesion molecules. And why do you want upregulated adhesion molecules? Also, actually, when the adhesion molecule is upregulated, there's another enzyme on the surface of the membrane that sheds it. And so what we were seeing in the blood in the proteomics was the shed form of the adhesion molecule, which is actually inactive, and it's just an indicator, a biomarker. So far, there's not really anything known about what the shed form does. It's really only active as an adhesion molecule on the membrane. And why does it go up? It goes up in order to allow leukocytes, so immune cells, it's like a sticky signal. It then allows the immune cells to bind to the cell membrane and then get into the tissue in order to, let's say you have an infection, in order to kill that infection, to treat it. And so that's really the role of increased adhesion expression. And so I looked under the literature, BCAM1, and interestingly, it's been shown that if you inhibit the ability of leukocytes to bind to BCAM1 with an antibody, you can prevent seizures in an epilepsy model in mice. It also is crucial, BCAM1, it's been shown in one region of the brain, not the one I studied, but the other region that's important for sensory formation, the olfactory bulb. BCAM1 inhibits neurogenesis. So with this knowledge then, if the factor goes up with age, I became interested in it and studying it and seeing if it's what is mediating the negative effects of old blood. So the first thing I want to do then is stain the brain, blood vessels in the brain, to see actually if the membrane-bound form goes up. And in fact, what I do see is an increase in BCAM1 in the blood-brain barrier, in the vasculature, in the hippocampus where neurogenesis occurs. So before I showed you guys the shed form in the blood, now we see the membrane-bound form is going up. Also, when I did parabiosis, what I saw is in the young mice exposed to old blood had this increase in BCAM1 expression. Then, interestingly, like I said, the issue with the parabiosis model is when you sew mice together, you cause more inflammation, there's a couple weeks of recovery. You don't know if they're sharing cells or it's just proteins. There's a lot of things that complicate the system. So then I just went back to my model where you just inject the blood plasma to show that it's really some proteins in the old blood that are inhibitory, and I see upregulation of BCAM1. So then, basically, we know that there's an increase in systemic inflammation. Now I showed you that there's an increase then in BCAM1 adhesion molecule on the cell surface. This then, at least in the literature, shows that it increases the ability of leukocytes, immune cells, to bind to the blood-brain barrier. So what I want to know is that do changes in BCAM1 with age have actual functional effects on inhibiting neurogenesis? Is old blood acting through BCAM1? We don't know. Just because it goes up as a result of old blood doesn't mean old blood works through it. I have to prove that. And so, as I said, I wanted to prove that. And so there's two approaches I'm taking. This is not published, it's all brand new. So I'm in the middle of doing this study transgenically, but so far what I've done is I've used an antibody, where I inject a neutralizing antibody in vivo, and all the antibody does is it doesn't prevent expression of BCAM1. What it does is it just prevents leukocytes from binding to BCAM1. So I'm sort of inhibiting its signaling that way. And what do I see? I see that in old mice that got the control, like IgG, it's not the real antibody, it won't do anything. You still get a lot of inflammation, so a lot of the IgA1 staining. But in the mice that I gave BCAM1, I did it over 3 weeks. I injected the mice 7 times total over 3 weeks, so every 3 days with the antibody, and then I just looked at the brain, and we see decrease in inflammation in the brain, as in decreased activation of the microglia.
So it does happen.
It's really exciting. I patented it, and we'll see if something comes of it. And then when I look at neurogenesis, and this is just an antibody again, systemic, so it's not invasive, so it's something that we could potentially use therapeutically. We also get a slight, not that much, but a little bit of an increase in neurogenesis, in proliferating PRG positive cells as compared to the control. But this is just in an old mouse, an old 18-month-old mouse. What I also really wanted to know, though, is can I add the antibody to an old mouse, but still I need to prove that it's due to old blood increases VCAM1, and because of that, it inhibits neurogenesis and induces inflammation in a young animal. And so in order to prove that, I turned to an immune deficient mouse. Why? Because, in order to do these studies where I inject mouse blood into mice, I have to kill a lot of mice. For just doing young blood versus old blood, I have to kill over, to collect the blood. A single mouse only has, a little bit of blood, so I have to kill over 100. It's kind of bad, right? So if I want to now increase my groups and add antibody, it's too much. So I turned to an immune deficient mice so I can use human blood. So this mouse, it's used in the lab in order to study most often cancer, because you can inject human cancer cells and it won't reject it because its adaptive immune system is deficient. But it still has an innate immune system, so it still has some leukocytes. So what I see, basically, is what I did is I injected either aged human blood, so from blood of humans older than 65 years, or just a control, with or without the antibody, with either the VCAM1 antibody or it's an IgG control. And I only had the antibody twice, just before I started injecting the blood, and then on the third day before I killed the mice. I also injected the BRGU, something similar, EG was called, like the same thing, in order to label proliferating cells. And what do I see? First, I see that in fact, VCAM1, it increases. Old human blood can induce, it's a little hard to see when you see an increase in VCAM1. And then with the antibody, it stays high. The antibody doesn't change the expression level of VCAM1. It just prevents the immune cells from being able to bind it. As you can see, it stays high here with the control, or when you add human blood in the antibody. Then, when I look at neurogenesis, you see, basically, I didn't show the PBS control, but you see inhibition of neurogenesis, a decrease in proliferation with the old human blood. But when you add the antibody, it rescues it. So here, what you see is you see an inhibition of proliferation, but then it's rescued by the antibody. So the antibody, basically blocking leukocytes' ability to bind VCAM1 in the cell surface, prevents the inhibitory effects of old blood. And then when I looked at inflammation, the same thing. You see that the human blood, just like the mouse blood, can induce inflammation. But when you add the antibody with the old blood, it prevents microglial activation. And so basically our model is this. With aging, you get increase in inflammation, which causes, let's see if I didn't do that, which causes VCAM1 to go up. That then allows more leukocytes to bind, and we're still not sure how, but this then causes on the brain side decline in neurogenesis and activation of microglia. What I haven't studied, and honestly it's because the techniques are just not available to do it now, I need to figure out how. If you have suggestions, let me know. But what is it that the brain endothelial cells are actually secreting into the brain? Or are leukocytes somehow getting into the brain? So far, we have no evidence. We've tagged the leukocytes with fluorescent molecules, and we've looked at the brain, and we don't see them going into the brain. So we think it's just binding, and then it's causing the endothelial cells to secrete something into the brain. We don't know what that is yet. I had كمان إشي بس يمكن I'll go past it. Just really quickly بسرعة. آخر شيء بسرعة. Just also what I see is that, I'm just going to say it really quickly, I won't explain it. But another thing I discovered in grad school and then we've patented is that if you take proteins and isolate them secreted by embryonic stem cells, so not adult stem cells, so throughout our body, throughout our life until we die, we have stem cells in every tissue. But these stem cells are only able to make that specific tissue. They're adult stem cells. But during development, you know, during development, at the beginning you have embryonic stem cells, and these make every tissue in the body. They're the ones that make the baby, right? Make the fetus. So if you isolate these cells and put them in a dish, and just take the proteins that the cells secrete, and you put them on adult tissue, they're like incredibly pro-regenerative. So I patented that. So maybe one day I'll make a medicine using that. But basically these are the kind of studies, if you want to look a little bit more, that I've published on this. And so I won't really go into this. But basically the way that these proteins act is that we see these, like I showed you guys already. I showed you TGF-beta signaling. And there are other pathways that regulate adult stem cell function that either go up with age or go down. And what these proteins do is they're actually causing them to all go down to young levels. And so by changing multiple pathways in the environment of the stem cell, that's how it's enhancing tissue regeneration. Right? So we know, like, the older you get, the ability to regenerate tissue goes down and down. But in the embryonic environment of the stem cells, you can make every tissue in the body. So if we can understand how to harness that for adults, we might be able to, cure many, many different diseases and prevent aging. And so basically that's kind of what I showed, is if you add the proteins, you increase proliferation. And that if you add it to injured muscle, the proteins, you get this enhancement of skeletal muscle, new fiber formation. Also, what I noticed is that you do this with mouse cells, and it also works for human cells. And so, what are my conclusions? Basically, I've shown you that TGF-beta and BMP signaling increases with aging, and in the local environment that the stem cells reside, and it inhibits neurogenesis, and it can actually induce inflammation in those regions. I've also showed you that old blood inhibits neurogenesis and induces inflammation through activating the blood-brain barrier and upregulating this molecule VCAM1. Approaches for rejuvenation include small molecule inhibitors, that's therapeutically relevant, also adding antibodies to prevent crosstalk of immune cells at the blood-brain barrier to prevent the negative effects. And then maybe even down the line gene therapy, because when I inject a virus, it does enhance neurogenesis. It's just more invasive right now, and that's a bit farther away. But there are people studying how to use gene therapy for that as well. So with that, I'd just like to thank my current lab. So right now I'm a postdoc at Stanford in Tony Wise-Cray's lab, which is actually a very big lab. And I collaborate on this project on VCAM1 with a scientist. She's actually from Germany. She's now also at Stanford. And then and another scientist with the immune regulation. I didn't show you guys that data. And then I have a few students at Stanford, studying their bachelor's, who work with me to help with this project, and who I teach. And then I had a student from the Netherlands come, and she got a master's with me from the Netherlands. And also my lab at Berkeley, I just said the lab that I was in, not specific names. And then the research was funded from the National Institute of Health. The U.S. government basically has a science branch. And then they've given me fellowships both for graduate school for my PhD, and then I got a fellowship now for this postdoc to support me. And shukran kthir. And
can I speak in Arabic? What I want to say here, not that. بشكل عام اشتغلتي انه هلا عندنا الرؤى الانفلاماتوري بروسيس انه الكاسكي اكيد بتفرز سايتوكاينز اللي حكيتي عنهم وبالتالي بصير عندك بروتينات اللي توكسيك انت لقيتيهم انه في الارض اكثر بشكل عام صح؟ فانت بتبريفنت السيجنالينج انا يعني انت صح يعني فهمت بمنع السيجنالينج انه هدول البروتين سينثيسيس هي المبدا. هي لا انا السؤال الثاني انه ستيم سيلز عندنا انا بعرف انه في البرين ما في ستيم سيلز عشان في جنريشن انه خلايا ما لا
في محلتين
بس محلين بس اثنين فاحنا لما نشتغل هيك بتمنع انه انا بمنع انه هدول البروتينات يزيدوا وبالتالي بوقف انه يصير عندك جنريشن لخلايا دماغ وهيك شيء. هلا بس هو التقنيه اللي انت بتصير فيها كمبدا هي بيولوجيكال لانه بيولوجيكال ثيرابي انه امنع البرو انفلاماتوري بروسيس اللي بتصير انه ما يصير سايتوكاينز وكيموتوكس ومش عارف ايه. فبالتالي انت بتاخذ ستيم سيلز من الامبريونات بصير انه تو انسرت
لا ما يعني
انا الفكره هاي ما
so there are people studying if you want to actually inject stem cells into the body. But this is why I don't really think we're far away from stem cell transplantations. Why? Because if the environment becomes bad, then even the stem cells that already exist are no longer able to work. So if you put old, new, even young stem cells into an old environment, because of these inflammatory proteins and unhealthy environment, they won't make new tissue. And the cells that they make will die. And so it's a much better, it's going to be more therapeutically relevant, okay, or more likely to make a therapy in order to make the environment of the stem cells more healthy, so that when they make new tissue, the tissue actually survives, and it also causes the stem cells that we already have to become activated. But there are people studying stem cell transplantation in order to, like you said, في محلات في المخ يعني فيش stem cells. إذا نحط فيه stem cells يمكن بصير neurons. في ناس يعني.
ما نستوب السيجنالينج هيك نشتغل على شيئين مع بعض. بتصير نيوروجينيسيس في stem cells أنت ما شفتيهم أصلاً معك. يعني لو بتصير
combine. You might be able to do that. You know, one thing is if you could take medicine to make your health, like the environment healthier, and then transplant new stem cells, you're more likely to have tissue regeneration and survival of it. So good question. Thank you.
I want to ask you if these techniques that you use work for younger people. I mean, young children with brain damage, maybe, or muscle damage. Are these techniques better for them?
That's a good question. With the embryonic proteins, the one that I showed you last, that will for sure work in even a young people. Because it's these proteins, some of them don't, once we finish development, we no longer express them in the body. So it's like we're kind of inducing changes that otherwise would not happen, so you could. With the TGF-beta inhibitors, maybe in young children with diseases, a lot of time these same pathways go up. So it's almost like premature aging. Like you see TGF-beta go up when you have cancer. You see it go up with children that have, brain damage, things like that. So in fact, it might be work.
Could we support that regarding the young blood and the old blood? Could we support that these results work with humans and encourage the people in their 20s or teenagers who may not geriatric people to treat their diseases?
That is a possibility. So right now, actually, the professor at Stanford I'm working with, he started a company just 2 years ago, and they got a lot of money to try two things actually. So right now, there's clinical trials going on at Stanford. Stanford has a hospital where they're injecting young blood into Alzheimer's disease patients to see if it can enhance their cognitive function and slow down the disease. We don't know yet if it's going to work, but there is some truth to the vampire tale. There is truth and yes, people are studying that. But what's more likely to become a therapy is going to be two things: finding what the rejuvenating young proteins are that you can inject, and seeing what goes wrong in old blood that you can inhibit with antibodies and small molecule inhibitors. Because it's not really, it's not ethical or it's not really feasible to be injecting, young blood into your body like throughout your life. But yeah, that's kind of, there are ongoing clinical trials now for that.
Sorry for being for this, but the more we age, the more like we we have one opportunity because there will be one mutation. Because like the telomeres in the like shorter and
the telomeres, yeah, you're right, there's telomere shortening.
This is of course that causes like mutations to occur, and as well for later to say if you want to die of anything, that's going to die of cancer at the end.
That's true.
Because of the mutations that occur. So body process should not be like find cures for
everything. That's
every cancer.
yeah, it's a very, very good point. So, you know, what our kind of thought is we don't, it's not that we want to make people live forever, it's just by having them age healthier, by making the brain healthier, by decreasing inflammation, because you're aging healthier, you prevent the onset of multiple diseases. Cancer, neurodegeneration, all those will maybe happen later or won't happen at all. You'll still get, you know, the telomere shortening really only happens in cells that are, most cells in the adult body aren't divided, right? Most are pretty quiescent. So you don't have to worry too much about that. In stem cells, they have telomerase in order to keep their telomeres longer. But there are, in the end of the day, you're going to have to take a multiple approach where you also enhance, like we know that with age you get oxidative damage, you get damage of the mitochondria. You're going to have to take things to help cell metabolism, to prevent inflammation, to get rid of. Also, you get an accumulation of, like you said, on the cells that are proliferating a lot, like especially immune cells will become senescent when they have their telomeres too short, and when they become senescent, they're not dead, they're not doing much, they start secreting pro-inflammatory cytokines. So we also need to find ways to actually get rid of these cells. And they've shown in studies, like when people study aging, if you get rid of these senescent cells, you can dramatically enhance the lifespan even of a mouse. So you want, in the end of the day, this is just for one therapy, and that's why, like I showed you guys, when I inhibited TGF-beta, I increased neurogenesis, but just a little. It was still way less than a young animal. And that's because there are multiple things changing with age to inhibit stem cell function, and to inhibit brain function. We have to tackle multiple things at once.
Because I appreciate and contribute to what you have done today for us. And I would really thank you for giving the recommendation for you in the presence of TGD and TGD and other series, not only if there will be no series in the medical field, cannot grasp or cannot conceive or receive all these details. I mean that TGD and the so next time we will conclusions or generalizations so that we can
understand a little bit more.
Around this.
No, definitely. I hope you guys can understand at least a little. I tried, but I will more. I'll make it a little bit more general next time. Thanks for coming.